Okay, guys, let's start. Assalamu alaikum. So today we are gonna finish this chapter about differentiation and stem cells, and today we're gonna talk about how the stem cell can be used for therapeutic purposes, and if it is at all possible to reverse the differentiation process, and can we do something in the laboratory that can be translated into clinic. So so we learned previously that normally during development, what happens is that starting from a single cell, which then divides into multiple cells, or after multiple cell divisions during embryonic life, it achieves a certain goal, and that goal is to specialize, to generate specialized cells which perform specialized functions. And this is the overall process is called development, and the mechanism it takes for cells to develop or for an organism to develop is through differentiation. So each type of cell specializes and differentiates into to perform a particular function. And now we're going to consider if this differentiation is reversible, and how plastic it is. And by plastic, I mean how dynamic it is. If a cell is normally during development, it goes on in one direction towards the differentiation, can we revert it and put it back to the state where it started? And if that is achievable, then it means that we can try to regrow the organs and replace missing tissues. And the biggest question is how to achieve this in a reliable manner, and to also achieve cells of a particular tissue, of a particular organ in sufficient numbers so that they can be actually used in a human being, in an adult human being. And that's actually the goal of the regenerative medicine. So we will start by asking this question first. Can we revert the pattern of gene activity in differentiated cells, and so that it mimics that in fertilized egg? And the short answer is yes, but it's a very challenging task to achieve in the laboratory. Still, I mean, so far we have achieved quite a lot of, there are quite a lot of advances in science and scientific methods, but still this task of redifferentiating or de-differentiating cells so that they mimic a more embryonic kind of cell, for example, mimic genetically and epigenetically to a fertilized egg or very early embryonic cells, that's a daunting task. So in principle, it is possible. And the reason why it's possible is because the differentiated state is not it's a stable state, but it's not a static state. So it can be reversed. So in this lecture, we'll talk about a few methods or few approaches that scientists have taken in the past, and they have, while they were studying this plasticity of the differentiation state. And the goal was to, so that the question they were asking was if this differentiated state can be reversed or not. And so they use different strategies. So for example, one of the strategies, or the first strategy that I'm going to talk about, is this nuclear transplantation, it's also called somatic cell nuclear transfer. So the basic question they were asking is that the changes that happen in the genome, because those are the changes that are reflected into the phenotype of the differentiated cells. So those changes that have happened into the genome of differentiated cells, can we reverse them? And what are the determinants, how we can reverse them? So they performed rather quite straightforward and simple looking experiment, although technically they were quite complicated and challenging at the time when they were doing these experiments. But but it was a simple idea. So they said that what if we let's take an oocyte or an egg, a fertilized egg, remove its nucleus, and then put a nucleus from a somatic cell in this fertilized egg. Do you guys understand what somatic cells are? What are somatic cells and what are germ cells? Anyone can tell me what somatic cells are? Guys? Ahmed Nakwi, are you there? Fatma Rahman.
Germ cells can produce, basically undergo meiosis and they can produce the leaves, and somatic cells are all the other cells of the body except the germ cells.
So the rest of the cells, yes. So the rest of the cells are somatic cells. And somatic cells, there are fundamental differences between somatic cells and the germ cells. So the germ cells, they undergo meiosis. The somatic cells, they undergo some of the somatic cells. Most of the somatic cells, they are end cells. They do not go into, they do not divide. But the rest of the cells, just a second, guys. So, yeah. Yeah, so this method that they developed or pioneered was known as the somatic cell nuclear transfer. And what they did was they took, for example, they used this xenopus type of a frog because the oocyte of a frog, it's a very big cell. I mean, it's and that's why they used it. So it has a very recognizable large nucleus, and it was easy to manipulate this oocyte from the xenopus in order to remove its nucleus and to add a nucleus into it. It was relatively easy to perform. So what they did is they took, for example, skin cells, cultured adult skin cells, or they took some other, for example, tadpole gut epithelial cells. So these cells, they are differentiated cells. So the skin cells and the epithelial cells, they are, as the name indicates, that they are differentiated cell types. So they took out the nucleus from one of these cells, either the skin cell or from the epithelial cell. And what they did with the oocyte, with the fertilized egg, was they treated it with the radiation. And we discussed last time that what happened when you irradiate cells. Anyone can tell me what happens when you irradiate cells? What happened to them? I think we discussed it last time. So when you irradiate cells, the radiation is a very high form of energy, a very high energy, and it destroys the DNA. It creates double-strand breaks, which are the most lethal lesions. So when you irradiate cells by either UV or by more stronger radiation like gamma rays, you basically irreversibly destroy the DNA. So then you have an oocyte which does not have any of its own nucleus, any of its own genetic material. So you introduce the nucleus or the genetic material from an epithelial or skin cell, which are two completely different cell types. And you introduce this nucleus into this enucleated oocyte. So what happened was, what the result was, they observed that the tadpole develops from this oocyte, although it did not have its own nucleus. It had received nucleus from or genetic material from somatic cells. And that discovery led to the belief that there are factors actually in the cytoplasm, which dictates actually the whole developmental regime. And whatever is inside the nucleus, it can be, it is quite plastic in nature. So it is very dynamic. It responds to the cues of the factors that are present inside the cytoplasm. Because as we have seen in this case, that when we put a differentiated nucleus into an egg, the whole thing normally develops into a tadpole, although it didn't go any further. And there were reasons for that, but at least it supported the early life, this somatic cell nuclear transfer. So this experiment clearly demonstrates that there are factors inside the cytoplasm, most likely transcription factors, that are enough to change the differentiation state of a somatic cell nucleus and convert it into a embryonic cell nucleus, basically. And for this discovery, actually, two guys, they received in 2012, they received a Nobel Prize in medicine and physiology. And for this discovery that that somatic cell nuclear transfer can create life, basically. In order to go further, what they did was to understand it further. They took, for example, again, took the xenopus as a model, and from the blastocyst stage of its embryonic life, when there are multiple cells, they took out some of the cells from blastocyst, take out their nucleus, and then implant it again into the oocytes one by one. And what happened was, the result was genetically identical frog develops. And we have seen previously that in this case, when you put a somatic cell from an adult or from late embryonic stage or after embryonic life, in the larval stages, when you take a nucleus from those at the later stages of life and put it back into the egg, the animal only develops until the tadpole. But if you take cells from the blastocyst stage, from the earlier stages of development instead of late adults, what happens is, if you put them back into an egg, they develop into the full animal. And that tells us that there is some degree of restriction that occurs if you, in the adult cells, that they, in a way that they cannot completely go back to the embryonic stage. But if you take cells from a nucleus from early embryonic stages of life and put them back into the oocyte eggs, they are able to fully de-differentiate or fully re-differentiate into embryonic stem cells and then develop into identical frogs. So these frogs were genetically identical, and this whole process was known as cloning. So one important undertaking that we need to understand here is that the genetic material after this in the either in the adult cells, for example, in the skin cells or epithelial cells, or in the slightly later stages of development, the genetic material is not irreversibly altered. So it is still plastic. You can revert it back to its previous state. So you can basically go back in time if you choose certain conditions. And one of those conditions was, if you take out the nucleus from a later stages of development and put this nucleus back into cells of a very early stage of development, then that nucleus or the genetic material in that nucleus, it can revert back to its initial state. So it's basically going back in time. And this is basically, this represents the functional plasticity of the genome. And the other important undertaking here is, a conclusion from these experiments is what I also discussed earlier, is that there is something in the cytoplasm of these oocytes that governs or that dictates the differentiation process. So it can differentiate or it can de-differentiate. So it's basically the factors, or transcription factors mainly. We will study that later. We'll learn that later, that this is basically dependent on transcription factors. But of course, a lot of other proteins take part into this whole process. But the important message is that there is functional plasticity in the genome, there, that it can go back to its previous state if you provide a proper stimulus. And that stimulus is provided by putting this nucleus back into an oocyte, which has all the cytoplasmic factors, or which has all the factors that are required to maintain this embryonic state of the cells. And then if you let them grow, they will develop into genetically identical organisms. You guys all must have heard about this famous sheep called Dolly. And that was also, this organism was also a result of a cloning procedure. And I mean, what they did was, scientists, they took cells from two different sheep. So they take somatic cell from one sheep, and then actually, yeah, so they take some somatic cells from one sheep, take out the nucleus, and basically put this into a blastocyst of a different sheep. And actually they took cytoplasmic and nuclear donors separately. It was more complicated than what we have learned here. So here in this case, there was only nuclear donor, no cytoplasmic donor. But in the case of Dolly, there was a nuclear donor and a cytoplasmic donor. So they took out the nucleus from one sheep and take out enucleated the nucleus from another sheep, another cell type, and then put this cell, which has cytoplasm from one sheep and the nucleus from another sheep, and put it in a blastocyst of a third sheep, which is a surrogate sheep. And then it basically led to the growth of a full organism, which was completely identical to its parents. And it's basically it's identical to its the nuclear donor, basically, because the genetic material came from the nuclear donor. And I mean, this was not a very simple kind of task to do or experiment to perform, and it is not always successful, as we will see a little bit later, that although, I mean, it is experimentally possible, but the success rate, especially in mammals, is extremely low. And because, I mean, the survival of this cell is extremely low, basically. And this is also a problem during this in vitro fertilization, that you basically take sperm and egg and fertilize it in vitro, and then you put it back into either the mother or the surrogate mother. But it there are a lot of technical issues with it. So with implantation, with the growth of this fertilized egg, there are a lot of things. So it's not always successful. The chances of success are very low, especially in mammals. There are many factors that prevent this from happening. But at the molecular level, the challenge was to understand if the differentiation was completely reversed at the genetic level. And one way to study that was to understand if all the epigenome, what we call the histone modifications and DNA methylation pattern, was it completely reversed or not. And then there is also a phenomena called imprinting of genes, which we might discuss maybe in the next lecture, or maybe not, but I will give you a brief idea. So what happens is, as you know, that so we receive genes from both our mothers and fathers, and some of the genes, they need to be silenced, and that is called the imprinting of genes. So for example, I can give you a very a prime example of this is the imprinting of genes on the second X chromosome in the female. So females have two X chromosomes, and one of the X chromosome is mostly silenced, and it is very highly compacted, and all the genes, most of the genes, they are silenced through different mechanisms. And that is basically so the paternal or maternal genes, they are silenced, and that also has something to do with the determination of sexual phenotypes, and also in the later age appearance of secondary sexual characters. So some genes, for example, in males, the female related genes, because we receive the full complement of genes. We also receive, for example, genes for estrogen. Males, for example, receive genes for estrogen, and females receive the genes for male sex hormones. But these genes are, or a lot of other genes, they need to be repressed in order for a particular sexual phenotype to appear. And this is a process called imprinting, and it starts already during embryogenesis. So one of the problems with this somatic cell nuclear transfer was that there was the epigenome reprogramming, or the imprinting of genes, was incomplete, and that was considered to be the most likely cause of failure in the success of this organismal cloning. And there were visible abnormalities that arose. For example, these organisms, so these especially in mammals, they had they die early. They had limb deformities, hypertension in, for example, large mammals like cattle. And small mammals, there were immune problems, etc. And in the case of this Dolly, it developed a very severe arthritis at the age of 5 years, and then she was put down at the age of 6 because she developed a lung disease, which was not related to its cloning procedure, but the arthritis was related to its cloning procedure. So there are obviously challenges with this method, but it is technically possible to clone a full human being, or clone a full mammal, or any organism for that matter. And recently, cloned blastocysts have also been achieved in primates and also in humans. So cloned blastocysts are this. So you basically introduce so you basically perform this somatic cell nuclear transfer with human oocyte, and let it grow into a blastocyst, and then you can use this blastocyst. We will further see what could be the potential uses of these somatic cell nuclear transfer. But yeah, so recently cloned blastocysts have also been achieved in primates and humans. So so obviously there are ethical concerns, which we will also discuss a little bit later, but there are practical and ethical concerns. It's a very technically challenging procedure, but there are obvious ethical concerns when you talk about human cloning. And yeah, but then some people believe that this can be utilized for therapeutic purposes or for studying the mechanisms of disease. which is a good idea, but then there are others which believe that if you make a blastocyst from a somatic cell nuclear transfer in human cells, and then if you kill that blastocyst to study the disease, it would be like killing a human being. So there are different point of views in this regard. But so far, the use of human blastocysts is very restricted, and I don't think that a lot of research has been going on. But the good thing is that the primates are a good substitute for research in humans, and there are less ethical issues, let's just say, with animals than with humans, although there are also animal advocates that believe that killing even a mice for cancer treatment is inhuman and should be stopped completely. But then there are others which believe that if you want to save a human life, then yeah, you can slightly justify, give some justification for using animals. So there are both point of views. Okay, so one method for changing the differentiation state of an adult cell is by this somatic cell nuclear transfer. So you just summarize it. So you take the nucleus of an adult cell and then put this into an enucleated oocyte, which could be mouse or human, or in case of Dolly, it was a sheep, and let it grow into blastocyst and let it grow into a full animal. And that would be this process. The whole process is called cloning, and this particular technique is called somatic cell nuclear transfer. And this is one of the ways to change, to study basically the plasticity of the differentiated state. And this experiment tells us that the differentiated state is not a permanent state and it can be reversed. Another way of reversing the differentiation is cell fusion. So for example, in case of human cells, or also in other animals, for example, other mammals, the oocyte is very small and it's very difficult to manipulate or enucleate the oocyte and perform this somatic cell nuclear transfer. So what people devise is they devised another way, because the idea is to expose one cell's nucleus with another cell's cytoplasm, which is what they did in the somatic cell nuclear transfer. So this idea can also work if they fuse two cells together. So that will also expose one cell type, one cytoplasm to another nucleus. And this was basically the experiment was performed by taking chicken red blood cells and fuse it with human cancer cell. And if you recall, we discussed last time that the red blood cell, the human red blood cells or mammalian red blood cells, they do not have nucleus, but because they have completely enucleated the nucleus. And that's why they took the chicken red blood cells, because that's a good model system for studying the cell fusion, because chicken red blood cells, they still keep the nucleus, although it's completely inactive, transcriptionally completely inactive. All the genes are repressed. They do not produce any RNA, but they still keep the nucleus. So the scientists asked this question, that if you fuse this chicken RBC with any other cell which has enucleated nucleus, remove the nucleus from where you have removed the nucleus, what would happen to the chick RBC nucleus, which is normally repressed, but if you expose it to a cytoplasm of a normal cell, or of a cell that has normal transcription factors and all that machinery. And what they observed was, very interestingly, the chicken RBCs starts expressing chicken-specific proteins when they put the when they fuse this chicken RBC with a human cancer cell. And So this actually tells us that the repression in the nucleus of the chicken RBC was not a permanent state. It was established in order for the RBCs to function properly, but it was not a permanent state, and you can induce de-differentiation, or if you can induce the reactivation of the genes. Another experiment that tells us that this fusion of two different cell types and two different species, it led to the reversal of the differentiation, was when they put the human hepatocyte into when they fuse human hepatocyte in with the mouse muscle cell. And so what happened was that normally in hepatocytes, these actin and myosin genes, or the proteins, they are inactivated. Because it's a hepatocyte, it's a liver cell. So in liver cells, you do not see muscle-specific proteins. But if you put it into a mouse muscle cell, which has all the machinery or all the transcription factors that were active in the mouse muscle cell, which were supposed to increase the transcription and expression of the muscle-specific proteins, those factors also acted onto the genes that were present, the muscle-specific genes that were present in the hepatocyte. So they were normally repressed, but when they were exposed to the cytoplasmic machinery or the nuclear machinery of the muscle-specific cell in mouse, the human muscle proteins started to being made. And this was yet another example of reversibility of the differentiation. And basically, it tells us that the gene expression program, it's not a permanent or a fixed program. It's a very dynamic program, and you can reverse it. And the other thing it tells us that it basically attests that or confirms that the reprogramming factors are present in the cytoplasm. For example, the transcription factors. So then we asked this question that, can this reprogramming, or if this reversal of the differentiation state, can it occur naturally? Anybody wants to chip in? So we discussed last time, there was this process called transdifferentiation. And transdifferentiation, the change, there is a change from one differentiated cell type into another. And now we're talking about adult cells, or cells in the adult organism. And this transdifferentiation is also an example of a reversal of a differentiated state, because in order for one differentiated cell to convert into a completely different differentiated cell, it first needs to be de-differentiated, and then it acquires the differentiation gene expression program of that other cell type. And there are some cases in both invertebrates and vertebrates that were observed where they saw this transdifferentiation. And let's take some examples from the invertebrates first. In C. elegans, it also has been shown in Drosophila, this transdifferentiation. But in C. elegans, what they've observed is that an epithelial cell, a rectal epithelial cell, is converted during the embryonic life. It's converted into a motor neuron, which then generated axonal processes and synapses, which are the characteristics of a neuron, of a nerve cell. So in this way, an epithelial cell is completely converted into a motor neuron, and this was normally observed in during the development in C. elegans. Another example was in jellyfish, where there are basically a successive transdifferentiation from starting from striated muscle cell into a smooth muscle cell. So the striated muscle cell, it was so when it is cultured in a extracellular matrix, the striated muscle cell keeps its or maintains its identity. If you digest extracellular matrix by different enzymes which degrade the extracellular proteins, these matrix proteins, then the striated muscle cell changed its phenotype and converted into smooth muscle cell. And then further on, it was further converted into a nerve cell. So, so this example basically highlights the importance of extracellular matrix, or the signals in the extracellular space that affect or that maintains the differentiated state. And if you take them off, then the cell changes its differentiation state. So it's both the factors inside the cytoplasm, but also in the space outside the cell, so what we call the extracellular space or extracellular matrix, because the tissues in the body, they grow, or in an organism, they grow in a three-dimensional space. They are not grown in a unilayer or monolayer. So there's always this extracellular or the microenvironment of the tissues of the cells, and that has a very important role in tissue growth and differentiation. So, and which is actually demonstrated by this example. In vertebrates, this transdifferentiation occurs very rarely, but it does happen in some cases naturally, and in others in pathological states. So let's consider first when it happens in a natural condition. So in this adult newt eye, so it's this salamander, it has a remarkable capability of regeneration, and we will study this regeneration in the next lecture, but and also study it a little bit more in detail. What are the signals and how this particular organism, it has this special property to regenerate all its organs, almost all its organs. So let's take an example of the lens. So if you remove the lens of this newt, it is able to regenerate this lens from the iris. So what happens is there are these pigmented epithelial cells, so when you remove the lens, they start to de-differentiate, and they change their shape from flat to more columnar epithelial cells. They lose their pigmentation, and they start differentiating into lens cells, which then secrete the lens. And I mean, of course, it's a gradual process, but this process is not actually dependent on the age of the organism. And it's not a limited capability either. So you can remove this lens as many times as you want, and it has the ability to regenerate it the same way again and time and again. So, but the important thing here is that the transdifferentiation occurs to a developmentally related cell type. So, for example, it was not possible for the cells in the iris, these pigmented epithelial cells, to transdifferentiate into a neuron, or transdifferentiate into a muscle cell, or transdifferentiate into a skin cell, because they would be from a different germ line. So, so here, for example, it's very important to understand that the stem cells, or whatever you can call them, the multipotent stem cells from the iris, or these pigmented epithelial cells, they had the ability or capability to differentiate into the lens cells, which is a developmentally related cell type. So if the regeneration needs to occur in, for example, in endodermal, cell types, they will only give rise to the endodermal tissues. They will not, for example, give rise to the skin tissues or the neurons, or which are derived from ectoderm. In humans, actually, there's this transdifferentiation was observed in a pathological state, and that was this is called Barrett's metaplasia. And what happens is that because of so there is a change in esophageal epithelium into a gastric type of epithelium. So the lower part of the esophagus that is in proximity to the stomach, this lower part is converted. So normally it has a stratified epithelium, but in the inside the stomach you have a columnar epithelium. So so this esophageal, the lower part of the esophagus epithelium is converted into a columnar type epithelium, and that happens because there is a problem in the gastric valve. So this the problem in the gastric valve leads to frequent regurgitation of the acid from the stomach, which destroys or which damages the lower part of the esophagus. And as a reaction, what happens is the cells, they turn to change their phenotype or their identity into a more gastric-like epithelium, because the gastric epithelium has the ability to withstand this very low pH of hydrochloric acid. And that's what is happening in this Barrett's metaplasia, that the lower part of the esophagus, it turns into a gastric type of epithelium in order to withstand this acid reflux or acid regurgitation. But this is also important to note here is that this type of transdifferentiation, so Barrett's metaplasia, it's an early stage of actually a cancer, because this conversion from one cell type to another, at least in human cells, it does not stay there at that point. So this conversion eventually leads to a gastric type of cancer in the lower esophagus. And then we have seen that experimentally, last time we discussed, or maybe a couple of lectures before, we discussed that the fibroblasts can be made to differentiate into muscle cells by expressing muscle-specific this MyoD group of transcription factors. And so if you take fibroblasts and start expressing the MyoD transcription factors, the fibroblast starts expressing muscle-specific proteins, and their phenotype changes into muscle cells. So this is also an experimental way you can change the differentiation state of a given cell type. Okay, so now we have seen at least two different ways to manipulate the differentiated state of the cell. So which actually now it's quite easy to understand that the differentiated state is a very plastic state, is a very dynamic state, and we can reverse it by at least, so starting with the somatic cell nuclear transfer, where you reactivate the developmental genes of a differentiated cell. So normally in differentiated cell, like we discussed earlier, that all the developmental genes or the stem cell related factors, they are repressed, and on the genes, the gene expression program that is active in that differentiated cell is only the one that is required for the function of that cell. So that's why it's called a differentiated or specialized cell type. But if you put this nucleus back into an egg, an oocyte, then there is reactivation of the developmental genes, and maybe switching off of that particular gene expression program that was specific for that cell type. And in this way, by the reactivation of developmental genes, the somatic cell nuclear transfer leads to the change in the differentiation state. And that was also we learned that happened. So this reactivation happened because there are factors in the cytoplasm, in the egg cytoplasm, which forces this reactivation of the developmental genes. And then we learned that if you fuse two different cell types, that would also affect or change the differentiation state of one of the cell type. And if one of the cell type or the recipient of the nuclear material is an embryonic cell, then the somatic cell nucleus is converted into, or somatic cell is converted into a pluripotent stem cell. But then there is a third way of manipulating the differentiation state of cells, and that is by inducing the pluripotency or inducing the pluripotent stem cell state. And so, like we learned previously, that there are factors in the cytoplasm that govern the differentiated state or undifferentiated state, for this matter, of any cell type. And from that, scientists deduce that there are probably a set of transcription factors that govern this change, or that can reactivate the developmental genes, and or that can lead to de-differentiation of any cell type. And while they were studying, or they were searching for these transcription factors, it was actually very surprisingly, it came out that there are only 4 transcription factors that are required to maintain this pluripotency-like state, or ES-cell-like state. And that was actually combined with this nuclear cell somatic transfer. This the guy who identified these transcription factors, this is Shinya Yamanaka, together with John Gurdon. They both received the Nobel Prize in Medicine and Physiology. So, like I said, that it was very surprising that only 4 transcription factors were able to lead to or completely reverse the differentiation state. Because one would expect that there are like 22,000 genes, and a lot of them, they are active during the embryonic life. But it boils down only to 4 factors that can lead to the change in the differentiation state, or that can reprogram any somatic cell into an embryonic-like stem cell with pluripotency features. And these 4 factors, they were NANOG or OCT4, either one of them, SOX2, KLF4, and together with CMYC. But there is a big caution sign here. The way you introduce these transcription factors is through some viral-based expression, or mainly retroviral-based expression. So, for example, Shinya Yamanaka performed this experiment where he took the skin fibroblasts and tried to overexpress, introduce these 4 transcription factors through retroviral-based expression. And so the transfected cells, they were treated with some drugs in order to keep the expression. So when you overexpress any gene within a vector, be it a retroviral vector or lentiviral vector or any other non-viral based vector, you always have this antibiotic selection, the mammalian selection gene. So, which means that these genes, so in order to test or in order to select the cells that are expressing these genes, which are part of this retrovirus, you add certain antibiotics. For example, let's take an example. There's this retrovirus. It has puromycin selection gene. So when you insert this into the cell, in any given cell, it not only expresses this particular transcription factor, but it also expresses the puromycin resistant protein. So when you add puromycin to the cell, so puromycin is an antibiotic, and usually antibiotics kill cells. So if you add puromycin to this mixture of cells, which some of them have received this retrovirus containing this, so only those cells will survive this antibiotic treatment that have this particular gene, because the ones that have this particular gene or this particular retrovirus, they will also have the antibiotic resistance gene. So this is a way to select cells for a particular gene expression. So when Yamanaka treated these, so it introduced these genes into the skin fibroblasts by retroviral expression, and then selected cells. Very few of the cells actually were expressing these transcription factors. So it's not easy to try to express these transcription factors. So cells, they turn them off relatively quickly because they are in a differentiated state. But of course, there are some cells, the transfection efficiency was quite low, and some of the cells, they did survive. And those that survive and after the antibiotic treatment, they were the cells that expressed these 4 factors, and then they changed their phenotype from fibroblast into the embryonic stem cells. The important thing here, and then these cells were then maintained in fibroblast growth factor, serum, etc., which are required for the culturing of embryonic stem cells. The important thing is here is that when you introduce any gene in a retroviral or a lentiviral vector, so these vectors, they have the property of integrating themselves into the genome, the host cell genome. So if you want to express some gene into this through this retroviral based expression system, you're basically changing the genome of the host cell. And sometimes it happens that it's extremely damaging for the cell because you cannot control where this retroviral is going to be inserted into the genome. And sometimes it is inserted at some crucial site that the cells cannot survive. And that's why the efficiency of transcription of the transduction with the retroviral vectors is very low. But of course, you have millions of cells, and some of the cells, they survive and they have a perfect set of insertion of the retroviral vectors and perfect expression. And these are the cells that then converted into embryonic stem cells. Another important thing here is that, so when you overexpress these factors, they are the pluripotency factors, but they convert the phenotype from fibroblast to embryonic stem cells, but then they are repressed. So they do not remain active in the embryonic stem cells. So they are the factors that are required for the conversion from, or the de-differentiation from an adult cell type into an embryonic cell type. But then there are so they turn on the other developmental genes that are required for embryonic stem cell phenotype. Yeah, so a caution needs to be taken while working with these retroviral based vectors. Yeah, so let's then discuss some of the methods that people use to derive this induced pluripotent stem cells. And we just discussed some of the problems with the viral vectors. Of course, the advantage is that they are much more efficient, and they, for example, a lentiviral, you can keep the expression for a longer duration of time. And the difference between retroviral and lentiviral is that you can use lentiviral vectors in the cells that are dividing, so that are replication proficient. Retroviral vectors do not work in the cycling cells, but you can use them in the end cells, for example, in muscle cells, etc. Then you have, let me see. So the lentiviral vectors, Sorry, I beg your pardon. So so the retroviral vectors can only work in the cycling cells. So they cannot transfect, for example, the end cells like muscle cells or nerve cells. So you cannot use them to transduce end cells. But the lentiviral vectors, they work in both end cells and the cycling cells. Because this is an important consideration when you trying to choose a cell line for making the induced pluripotent stem cells. Then you have, and the disadvantage is we discussed earlier that these vectors, the retro and lentiviral vectors, they integrate into the host genome, and you cannot control where they integrate, and that's why they can lead to damaging or deleterious effects on the cells. Secondly, the lentiviral vectors, they have these long terminal repeats that remain active in the host genome, and they have the ability to recombine, and they can change places. There are transposons that move these long terminal repeats from one place to another, and they can induce some, or they can change the expression of some genes, and it may lead to cancer or some other diseases. So care must be taken, because this property of integrating into the genome is both a good property in a way that then you achieve a stable kind of expression, but on the other hand, if this integration is at a wrong place, it will have quite deleterious effects. Then you have DNA plasmids and transposons. So you have DNA plasmids that are not viral based, and of course you can use them, but their efficiency is quite low because they are not stably integrated into the genome. So their expression will last, where it will subside very quickly. Then you have transposons based vectors, and transposons based vectors means that that you have certain enzymes that basically cut and paste the cargo DNA. So if you provide a gene, so if you clone a gene into these transposon based vectors, this the transposition occurs. So there are transposases that basically take your gene and insert it into at a specific place into the genome. But again, I mean, the efficiency of the transposons is also quite low. So then you have these non-DNA methods. So you can also introduce the RNA of these transcription factors. You can also put these transcription factors as directly as per recombinantly purified proteins. But I mean, again, the survival of the RNA, RNA is a very sensitive molecule, and it's not easy. I mean, it gets degraded very quickly. So it's not easy to work with RNAs. Transcription factor proteins, that could work, but then it also depends on the size of the protein, and big proteins, they have difficulty crossing the cell membranes. So yeah, it's also more challenging. Then there is also a possibility that you screen, for example, drugs that activate the expression of these transcription factors. That's a very long-term kind of goal, but it's an ideal situation, because if you can use the small molecule inhibitors, they could be very specifically turning on these 4 genes. But yeah, it's a very daunting task. It's not easy to identify small molecules that can induce the expression of these pluripotent stem cells. So, I mean, all these methods, they have their advantages and disadvantages. And as a molecular biologist, I think you all should know these are very practical approaches that people use in the laboratory these days. And you should know the ins and outs of these, or advantages and disadvantages of these, especially the viral vectors. I mean, these retroviral and lentiviral vectors, they have been used very extensively in the molecular biology labs throughout the world. So yeah, so it's important to understand how they function and what are the different advantages, disadvantages, or risks associated with it. Because I mean, in the lab-based setting, of course, if you are not injecting any material into a patient or into an organism, then it's okay wherever the viral vectors they are integrated. But if eventually you want to introduce these cells, iPS cells, therapeutically into an organism, into a human being, then of course these things matter quite a lot. So yeah, so these to sum up, this induced pluripotent stem cells, so you can generate them by introducing 4 transcription factors that when expressed, they de-differentiate the cells, and then their expression turns off, and they induce the expression of developmental genes. And in this way, you can convert any adult cell into a pluripotent stem cell. An important thing is that they are pluripotent stem cells, so they can be differentiated into any cell type. And then again, they will also have the problems that are associated with pluripotent stem cells, which we'll also encounter later, that they can grow into teratomas and cancers, which is a side effect for these pluripotent stem cells. Okay, so let's take 5 minutes break and then we will resume. So let's come back at 11:15. Okay, we are back. आप लोग हैं या बस कंप्यूटर ऑन करके चले गए हैं?
We are here, sir.
अच्छा, सारे तो नहीं लग रहे मुझे. Fatima, are you there? Ania, Kanza,
Yes, sir.
Marich, Savera, Ahmad. मैं ख्याल है आप 3 लोग ही हैं बस, बाकी 3 नहीं हैं. All right.
Yes, sir.
Are you following the lecture? How is it going? Can I have some feedback? Guys? समझ आ रहा है आप लोगों को कुछ?
आ रहा है.
हाँ. कोई क्वेश्चन वगैरह भी पूछ लिया करें कभी कभार गलती से, कि सारी चीजें बिल्कुल सही समझ में आ रही हैं. ऐसा तो मुश्किल है वैसे, लेकिन all right, but don't be shy, just stop me if you don't understand anything, and I can explain it again. All right, so we were discussing some cell replacement therapies, and the goal is to regenerate the damaged or lost tissues. And in this context, we discussed that how we can utilize this plasticity of differentiated state and revert it into previous differentiated state or undifferentiated state, and use it for therapeutic or disease studying purposes.
So you imagine you have a damaged or diseased tissue, and there is an adjacent normal tissue, and then there are different strategies that you can use to recreate the normal tissue in place of this damaged diseased tissue. So one strategy is that you can use drugs that induce the differentiation of this stem cells in this normal tissue so that they can produce replace this damaged tissue. They can produce new tissue that replaces the damaged tissue. On the other hand, you can also take stem cells or generate embryonic stem cells either by somatic cell nuclear transfer or by cell fusion or by inducing pluripotency into the somatic cells. And then you differentiate them into this particular cell type and then replace this tissue. But this is actually a very challenging task. It has been achieved for some tissues that they took stem cells and induced the differentiation of a particular tissue and then replace it in place of the damaged tissue. And another thing is that you take these stem cells, induce the differentiation, and then put them together with scaffolds, and then put them back into the normal, next to the normal tissue or next to the dead tissue. So what does the scaffold means here? So scaffold is, we discussed earlier that the cells in tissues or organs, they do not grow in single layers. So they always grow into a lump, or they always grow in a three-dimensional space. And in this three-dimensional space, there is a extracellular matrix, which is mainly composed of proteins, but it also has different cells as well. For example, fibroblasts is one of the connective tissue cell type that is present in the extracellular matrix. And actually the fibroblasts, they secrete this extracellular matrix. And this matrix is very important in the three-dimensional organization of any tissue or organ. And without this three-dimensional organization, you cannot achieve a tissue homeostasis. And we have seen previously that transdifferentiation is also, or a differentiation state of some cells that are dependent on the extracellular matrix. For example, we've seen that previously that the muscle cells, when the striated or skeletal muscle cells, when they are grown in extracellular matrix, they retain they stably maintain their differentiation state. So if you remove the extracellular matrix by digesting the proteins of extracellular matrix, the skeletal muscle is differentiated into, or changed or transdifferentiated into smooth muscle cell, which is completely different in shape and properties and functions as well. So we will discuss a little bit more about these scaffolds, but let's take an example of this macular degeneration, where there is a permanent and irreversible damage in the retina. And so the scientists, they try to repair this damage by retinal pigment cells that were induced from human epithelial stem cells. And so what happens is that the scientists took the human epithelial embryonic stem cells, and they induce them into the retinal pigment cells, and then put them, or surgically implanted these. So first they grew these retinal pigment cells in the lab, and then they collected these cells and then surgically implanted them into the eyes. Sorry. And what they observed was that these retinal pigment cells, they actually partially cured. They were, first of all, well tolerated with the patient, and they partially cured the visual acuity, so improved the vision of these patients. But of course, I mean, it's important to consider that if you take embryonic stem cells from from a different person, and even if you try to induce the particular type of differentiation and induce them into a particular cell type and then surgically implant them into another person, what would happen? If you take cells from one person and inject it into the other, what would happen? Anybody can tell me? Guys? It's a high school level question. So what happened if you, what is the major risk when you transplant bone marrow, for example, from one person into another? What is the biggest risk? What could happen? Or what is the biggest
Rejection.
challenge? Yes, so the transplant
Rejection by
rejection.
the recipient.
Yes, so the immune rejection by the recipient cell. So this could definitely happen in the same way this could happen in this case. So if you take out epithelial embryonic stem cells from one person and induce them into a particular cell type and then inject it into another person, you will end up having this problem that it might be rejected by the recipient. So, but at least in this case, it didn't happen, which was surprising and good for the patients. And these results basically encourage the scientists, and they are actually doing more trials in this particular disease. So we discussed earlier that the biggest risk apart from the rejection, I mean, you can alternatively, you can take the embryonic cells from the same person or related person. Or for example, I told you that in some Western countries nowadays, when a baby is born, they keep this part of the umbilical cord because it is a source of perinatal stem cells and embryonic stem cells, and they keep it. And if, for example, in later life, if the child or when it's grown up has some disease, when he or she needs some of this kind of treatment, for example, induced pluripotent stem cells, so they can use his or her own embryonic stem cells and then try to induce differentiation of a particular type. But if you take cells from somebody else, then you will always have this problem of rejection by the immune system. The other problem with this embryonic stem cells is that since they are embryonic, so they are pluripotent stem cells. And the pluripotent stem cells, they have the ability or capability to differentiate into many cell types, and that is, on one hand, that's a good capability, that's a good property, but on the other hand, it can also lead to these tumors called teratocarcinomas. So they are called teratomas, so they can be both benign and malignant. So the malignant one is called teratocarcinoma. And these are embryonal tumors, and they have tissues from all the three germ lines. So yeah, so they have nerve cells, they have muscles, and they have epithelial cells. So they are different varieties of these teratos. So teratocarcinoma is an epithelial teratoma. Yeah, so, and this was demonstrated very nicely by this experiment where the mouse embryonic stem cells, they were grown in culture in a monolayer, as you can see here, in a petri plate. And then it was either injected into the normal blastocyst in the inner mass of the blastocyst, or it was injected under the skin of an adult mouse. So what happened was that in this case, on the left side, when it was injected, when the mouse embryonic stem cells were injected into the blastocyst, this blastocyst, it developed into a chimeric mouse, because so these cells are from a different strain of mice. So for example, judging by the color, you can see that they are from a different strain of mice, and when they are injected into a different and a second strain, this blastocyst develop into a chimeric mouse. So it has properties of both this kind of cell and this kind of cells. But when these embryonic stem cells are injected into the skin of the adult mouse, what happens is they lead to the formation of teratocarcinomas or teratomas. So it's quite dangerous to use the embryonic stem cells. Of course, when you use the embryonic stem cells, you do not normally inject the embryonic stem cells directly. So first you induce differentiation into the embryonic stem cells, or try to induce differentiation into a particular cell type, and then you use that particular cell type of a particular tissue for therapeutic purposes. And so the important consideration here is that before, for example, therapeutically injecting it into a patient, it's very important to make sure that there are no undifferentiated cells that are still present in that mass of cells that you are injecting. Because, so like I said, that so you take embryonic stem cells, you differentiate them into a particular cell type, and then you take the differentiated cells and inject it into the patient. But imagine if there are there are still undifferentiated cells present in this, among this population of differentiated cells. So what would happen is, when you inject this heterogeneous population of cells, which contains both differentiated cells and the embryonic type undifferentiated cells, they go into a patient, and then these embryonic type cells, they had the potential to grow into teratomas. So that's a big risk. So this therapeutic use of stem cells or differentiated stem cells, it requires very stringent selection procedures to ensure that you only have the differentiated cells and no undifferentiated cells. So, like we discussed earlier, that the problem with the embryonic stem cells, although they are both embryonic stem cell and induced pluripotent stem cells, they are both pluripotent, but the problem with the ES cells is that they can be rejected if they are not from the same person, and which is usually not the case. They can lead to immune rejection and cause severe graft versus host disease as well. But the good thing about induced pluripotent stem cells is that they are induced, and they are induced in the laboratory. So you can take cells from the same person and induce them by overexpressing these 4 transcription factors, and then you can try to redifferentiate them into a particular cell type. So first you de-differentiate into iPSCs, and then you use these iPSCs to redifferentiate into a particular cell type, whatever you would like to transplant. But then the good thing about iPSCs is that they are from the same individual, so they will not lead to any immune rejection. But then there are, of course, other problems with iPSCs, because the way you are making them, that may cause problems. For example, we discussed earlier that most of the time they are made by retroviral or lentiviral vectors, and they have their inherent problems. Another thing that needs to be considered here, and the difference between ES cells and the induced pluripotent stem cells, is that, or the difference between the ES cells made by the somatic cell nuclear transfer and the iPSCs. So both of the cell types, they are from the same individual, so they will not be a problem with the immune rejection. But between these two types, the cells made by somatic cell nuclear transfer, they are of better quality in producing a particular differentiated cell type. Why? Because they are genetically very similar to the embryonic cells. While the induced pluripotent stem cells, we also learned earlier that there is incomplete reprogramming. in the iPSCs. Although some degree of reprogramming problems also occurred in somatic cell nuclear transfer, which we learned in the cloning of Dolly the sheep, that or the other animals that are produced by cloning, is one of the reasons of its low rate of success was because of the incomplete reprogramming at the epigenome level. But in induced pluripotent stem cells, this problem is even more severe, that the genetic makeup, the epigenetic landscape is it's not it's different from the embryonic stem cells. So it still has some imprinting issues, and it still has a lot of genes that are not fully reprogrammed, or that are not completely silenced or activated as it should be in the stem cells. Then, of course, there are, in the somatic cell nuclear transfer, there are, or the process that you call cloning, there are ethical issues, which I think we have covered earlier. So the alternative to somatic cell nuclear transfer is basically either transdifferentiation, or you use induced pluripotent stem cell, or you use adult stem cells. Yeah. So the ideal thing is, of course, adult stem cells. And yeah, because so the problem with adult stem cells is that they are not pluripotent. But the good thing about adult stem cells is they can be taken from they are from the same person, so there will not be any immune problem. And they have been shown for many decades that they have been used for immune disorders and cancers, where people have been receiving bone marrow transplants for different diseases. And we have also seen, last time we have seen this patient with this epidermolysis, junctional epidermolysis bullosa, where he had this detachment of the skin problem throughout his body, and then it was replaced by the stem cells, or stem cell grown skin. So adult stem cells is definitely the best you can get, because it undergoes the minimal amount of manipulation, while the other methods, all the other methods, even if it's somatic cell nuclear transfer or induced pluripotent stem cells, or using embryonic stem cells, they all go through quite a lot of technical experimental stuff, and that creates a lot of problems at every step. And we have seen also last time that the single stem cells from the crypts of small intestine, they can grow to form organs. I mean, they usually, what's happening inside our gut is that cells at the bottom of the crypt, they are continuously growing, and they grow into form these villi and line the villi, etc. And it has been shown that a single stem cell in the laboratory, it has been shown that a cell from crypt, it can go into form organ, the full-blown small intestine. And this is what happened. This technique to grow a single cell into a full-blown organ, a three-dimensional organ, this is called the organoids. And so in organoids, what you do is you take stem cells and differentiate them into specific types under specific culture conditions. But the important thing is that that you do this in a 3D space. So you provide a three-dimensional meshwork, or a three-dimensional matrix, where this, which can be both artificial or natural. And you provide this 3D space, and the cells, they grow into this 3D space, and depending on which cell type you are growing, or which stem cells you are growing, for example, if you're growing the stem cells from the small intestine or from the crypt, bottom of the crypt, they will only grow into the small intestine. They will not grow into blood cell or any other cell, because as we discussed earlier, that the stem cells, they are adult stem cells, they are multipotent, and they are already determined to follow a particular lineage. So if you take, for example, a hepatocyte precursor or hepatocyte stem cell, they will only grow into liver, but they will not grow into intestine. But the important point with organoids is that, or the good thing with organoids is that you can grow the whole organ, of course, in a miniature format, but you can grow the full organ starting from single cell, and provided that you give them a 3D culture environment. And so in the intestinal organoids are the most successful and advanced organoids, and they have been shown to be grown from both tissue biopsy and from the induced pluripotent stem cells. So it has been shown that these organoids, they replace, for example, the artificial bile ducts were generated by growing epithelial cells from the healthy ducts and the tubular scaffolds, but also in case of the intestine, it has been shown successfully that it can be used therapeutically. So what happens is, so how does this organoid grow? So you take a single intestinal stem cell from the bottom of the crypt, and you let it grow. And when it grows, of course, it's the stem cells, as we learned earlier, that they have this asymmetric cell division. So when it grows, it divides from 1 to 2 cell. It always gives one cell of its own kind, and the second cell, which is a more differentiated or a precursor of a differentiated cell. And so in this way, this single intestinal stem cell, it gives rise to a population of cells which contain both the stem cells and spinat cell, goblet, enterocyte, and enteroendocrine cell. Anybody can tell me which of these cell types they are responsible for absorbing the nutrients from the gut? So which of these 5 cell types is responsible for absorbing the nutrients from the gut? Guys? Okay, I guess everybody is sleeping. So it's the enterocyte that is required for the absorption of the nutrients. So yeah, so from a single intestinal stem cell, you can grow a full-blown intestine, provided you give a 3-dimensional matrix, which it could be a matrigel or it could be a natural matrix, extracellular matrix. So there are several uses of organoids. I mean, the major use is, or the idea behind growing organoids is to develop organs inside the laboratory, and so that they can be used in regenerative medicine. But on the other hand, they can also be used to study the pathophysiology of a particular disease, because you can make modifications into the cells, into the stem cell at the start of the stem cell, and then you can create a disease model of a particular disease. For example, if there is a disease which has only a single gene mutation, you can very easily generate an organoid model which has all the cells that have this mutation, and then you can study this particular disease, what happens with this at the cellular level, at the molecular level. And then you can also test different inhibitors, different drugs, how this organoid, how this mutation respond to the particular drug. And this is actually has been used quite extensively in the cancer treatments, because organoids can be made from the cancer patient, and then different drug combinations or different drugs can be tested on that particular organoid, and it responds very reproducibly and exactly in the same way as that person's own tumor tissue would respond. So it's a very nice way of testing different drugs. Of course, it takes some time to grow this organoids and then to test different drugs, and but there is a way to do this. So it's also been shown that the ES cells can be developed into a mini brain and into the embryonic intestines, etc. It's a more complex procedure, but organoids have now been not very commonly, but they've been made and they've been incorporated into the therapeutic procedure of different diseases. So the important thing is that, and which we do not yet completely understand, is how the cells, they self-organize, how the cells are conscious about their self-organization. And this is the key to the 3D development or the organization of a particular tissue into an organ. Because somehow, and it's most likely related to the factors that are present in the extracellular matrix. As we have seen previously in the example of the striated muscles, that in the presence of extracellular matrix, striated muscles keep or maintain their identity. But if you digest the extracellular matrix, they transdifferentiate into smooth muscle cells. So this is an example that 3D environment exerts, how 3D environment exerts its effect on differentiation state of a particular tissue. So, but of course, there are other factors inside the cells that understand or that recognize this, that are conscious about this organization into a 3D organ, which is so far not very really fully understood. And finally, we will consider the generation of insulin producing pancreatic beta cells, which is also a prime medical target because of hundreds and millions of people suffering from the diabetes. And so as you probably have all known that or learned previously, that insulin that is required for lowering down the blood glucose level, and that is the main problem in the type 1 diabetes mellitus, that the insulin producing cells, the beta cells of pancreas, they have run out. So they are completely destroyed because of some autoimmune problem. And that's why these patients, so the type 1 diabetic patients, they cannot produce any insulin, and that's why they have high blood glucose, and it's all of its secondary problems then. So the kind of prime medical target for cell replacement therapies is to remedy this loss of pancreatic beta cells. And it wasn't a very easy task, but and previously what people have done was to use the cadaver pancreatic tissue and transplant it into patients. But though it had some success, but there were a lot of immune problems and rejection problems. And also that the transplant tissue is scarcely available. It's not very easy because donors are not very frequently available. So so then the scientists, they decided to test this idea of developing pancreatic beta cells from either from the embryonic stem cells or from the induced pluripotent stem cells, because that would provide a kind of a long-term solution for this type 1 diabetes mellitus. Yeah, so like I said, that it wasn't a very straightforward protocol to differentiate stem cells into the pancreatic beta cells, the endocrine part of the pancreas. But it was so they started with human embryonic stem cells, and then try to use conditions that would convert them into the endodermal cells expressing this FOXA1 transcription factor. And then this FOXA1 transcription factor, it expresses, or it forces the expression of this PDX1 transcription factor, which is a pancreatic beta cell transcription factor. But the problem was that during this induction of differentiation, only very few cells, so 5% of the cells eventually end up expressing the PDX1 transcription factor. So then the scientists decided to increase this population of cells expressing the PDX1 transcription factor. And they searched 5,000 compounds, small molecules, and finally found out one molecule or one compound that effectively increased the transcription of this PDX1, and that was indolectin-5. And together with indolectin-5 and fibroblast growth factor, the cells, they started expressing, 45% of the cells started expressing the PDX1. So they increased the efficiency of transcription of PDX1 from 5 to 45%. 9 times more expression was achieved by adding this indolectin-5. So, and eventually they had some success, and the cells started producing the insulin. Although now there are clinical trials running on this kind of therapy, and it's still not readily available. But there are, I mean, this is a very good news, because it basically proves the principle that basically stem cells can be used to regenerate lost or damaged tissue. Yeah, so actually the human pancreatic progenitor cells derived from these pluripotent stem cells, they are in, like I said, in clinical trials for the cell replacement therapy. Then there were also attempts to make pancreatic beta cells from the iPSCs that were generated from fibroblasts. And then there were some attempts of somatic cell nuclear transfer, induced transdifferentiation of liver cells. I mean, people have tried all these different strategies, but I think, like I said earlier, that adult stem cells, they are the answer, because there are much fewer side effects of adult stem cells, and that is yet to be identified in pancreas. So far, the best treatment available is the human embryonic stem cells that are then induced to differentiate into the pancreatic beta cells. There is also an attempt to induce the transdifferentiation of exocrine pancreatic cells into endocrine by overexpressing the PDX1 transcription factor into the exocrine pancreas. But again, there are inherent problems when you overexpress a particular gene through viral vectors or through other means, and there are other problems. So there are many different ways to generate replacement cells in case of pancreatic cells. So here, there are different approaches are listed. So for example, you can take a skin fibroblast or either follow a somatic cell nuclear transfer in unfertilized egg, let the blastocyst grow, and then you take the inner cell mass of the blastocyst, grow into a Petri plate, and because then it gives human embryonic stem cells. On the other hand, you can induce the pluripotency directly into the skin fibroblast by transfection with OCT4, SOX2, KLF4, and CMYC. And these are and then let them grow in a Petri plate. And these are the induced pluripotent stem cells. So either you take in iPSCs or ES cells, and then you go through this 9-day lengthy and laborious protocol. and overexpress the PDX1 cells, which then produce insulin. These are two different ways, and then you can also, of course, transdifferentiate, or let the transdifferentiation, or use the transdifferentiation procedure in vivo. So you can change the adult exocrine pancreas or the liver, and so you transfect the transfection of organs with PDX1. Like I said earlier, that you can overexpress PDX1 into these cells, and then they start expressing the insulin. Or you can also use the PDX1 as a protein and add it to the cells, to the exocrine pancreas cells or the liver cells, and that will convert them into beta cells, the endocrine pancreatic cells. And another approach for the cell replacement therapy is to grow organs of one organism in another for subsequent transplant. This is, I think, quite futuristic approach, and not a lot of trials have been done on this, but it is, of course, theoretically possible. Also has a lot of ethical issues, because of course you have to kill the other organism. Yeah, so the ideal approach, I would say, for cell replacement therapy is organoids. But then again, where would you get the stem cells for organoids? That's another debate. So that's another problem. So I mean, all these different cell replacement therapies, they have their pluses and minuses. They have their advantages and disadvantages, and people are constantly or continuously working on them. But like I said earlier, that the best approach is to find out adult stem cells and let them grow into organoids, which is not an easy task, but that's the best approach, because I personally believe that all the cure of a human body is present within, or most of it is present within. If you try to cure it from outside, you will have a lot of problems. So yeah. So yeah, so today we considered different aspects of the of how we can change the plasticity of the differentiated state, and then we studied different mechanisms where we can use our knowledge about the differentiation, or how the differentiation is maintained, and then use it to reverse the differentiation process. And so there was this somatic cell nuclear transfer and the cloning approach, and then you have this cell fusion, induced pluripotent stem cells, and finally we discussed about organoids. All right, so that was today's lecture. If any of you guys have any question, please go ahead. Nothing? All good? All right, so then see you on Thursday morning. Okay, Allah Hafiz.